Hi, everyone. Recently, we studied evolution and adaptations, and we looked at some of the evidence that supports evolution as a theory, like fossils, and how people had used fossils for a very long time to see that organisms did have similarities throughout time, but also accumulated these changes that made them different from one another as we get to more advanced layers of the fossil record. We also looked at embryological data and how different species develop as embryos and retain a lot of the same characteristics throughout that development. And the more characteristics that they keep over time, then the more closely related those species are assumed to be. But we also look at the best line of evidence, which is biochemical data, DNA, RNA, and protein sequences, things that you can sequence. Because these sequences have been passed down from parent to offspring with these minor changes, we can look for these very slight changes between species and see how these mutations were passed down that caused all of this change. So now we're going to look at how organisms are classified and how these relationships are expressed. The field of biology that deals with classification is called taxonomy, and the scientists that sort these organisms into groups are called taxonomists. The original taxonomists just basically looked at physical characteristics to put organisms into groups. For example, mammals have fur, so they should all be grouped together. Birds have feathers, they should all be grouped together. But today's classification is done with a few more lines of evidence like we've looked at, not just physical characteristics, anatomical data, but also embryological development, biochemical analyses, your DNA, RNA, and protein sequences. And these are all used to reflect evolutionary relationships. So we're going to look at how this classification system works. But basically, you can see that we have different groups that are very inclusive. They have lots of different organisms that belong to them. And as we move down this classification system, we become more specific in the characteristics that we're looking for until there's only one species that fits those characteristics. So for example, here, we start out with the kingdom Animalia. All of these organisms are animals. If we go to the phylum Chordata, and there's different phylums for different animal groups, the chordates have spinal cords. So if we notice all of these organisms now have spinal cords, we have removed the sea star. It belongs to a different phylum called the echinoderms. If we move down to the next level of classification, we're in the class, and class Mammalia has fur. So all of these organisms have fur and a spinal cord, and they're all considered animals. So this weeds out the snake. The snake does not belong to the mammal group. If we move down to the order Carnivora, then we are looking for things that are considered carnivores. And I know the panda doesn't belong here necessarily. It's not a carnivore, but it is closely related to these bears, and so it does go into this group. There are some minor exceptions that you'll notice. But anyway, the carnivores do not include things like squirrels, not rodents. So we eliminate that from this level. We go down to the family Ursidae. These are the bears. The fox is not a bear, so it gets eliminated. The genus Ursus are the true bears, the American bears, so we eliminate the panda. And species Ursus arctos is your grizzly bear. And so we have eliminated the black bear because only the grizzlies fit these characteristics. So organisms are classified by different levels, like we just saw. The most inclusive level is the domain level. There are only 3 domains, and we'll look at all 3 of them here in just a minute. And we can go from the domains, break those into kingdoms, phylums, classes, orders, families, genus, and species levels. So you can remember this by the first letter of each word. Dumb, king, Philip, came, over, for, good, spaghetti, sushi, salami, whatever you might like that starts with S. So just a reminder of what a species is. I know we've talked about this a lot. Organisms that have very specific characteristics in common, and they are so closely related that when they breed, their offspring can still produce more offspring. They are viable. So a horse and a donkey, not the same species, because when they are bred together, we get a mule, and mules are sterile. They do not make more mules. The most inclusive level, like we said, is the domain level, and there are three. So we start with a common ancestor called the LUCA, last universal common ancestor, and it moves along, staying fairly the same for a while, and then it breaks away into a couple of different groups. So there's a mutation. We have the bacteria, and we have the archaea, and the archaea and the eukarya are assumed to be more closely related than the bacteria and these other two. So the archaea and the eukarya have a divergence as well. So these are our three domains. And here we can see that the domains are broken into kingdoms. So each domain has at least one kingdom. Bacteria and archaea only have one, the eubacteria and the archaeobacteria. Eukaryotes have four kingdoms: plants, animals, fungi, and protists. And we're going to look at each one of those as well. We'll start with domain bacteria. They have only one kingdom, eubacteria. Eu means true. We've seen that before. So true bacteria. They are single-celled. Sometimes they will form a cluster, but they do not work together. They are individual cells that can be separated and live just fine on their own. But due to proximity, sometimes they'll just kind of hang out in a group. They don't necessarily move away from one another once they divide. They are prokaryotic. That means they lack a nucleus. Remember, pro means before and karyo means nucleus. And they have a cell wall made of a sugar compound called peptidoglycan, which is targeted by most antibiotics. They poke holes in this peptidoglycan so that water will fill in the bacteria and it will burst the cells. So that's how an antibiotic works, most of them anyway. Now, scientists do disagree on which came first, the bacteria or the archaea. And there are many scientists that fall into each group. Most groups lead toward the bacteria or being the first, but there is some line of evidence that says it could be archaea as well. Now, bacteria, we'll look at their structure. They have three outer layers. They have a capsule, which is this slimy layer that they can slip out of if they start to be captured. So if an amoeba is closing in around them, then they will slip out of that capsule and they'll get away. Inside of that, they have a cell wall, again, made of peptidoglycan, very tough sugar complex. And then on the inside here, we have the cell membrane. Remember, all cells have a cell membrane. So this membrane is the first living layer that we're going to get to, and it's going to secrete these other two layers for protection. Some bacteria have a flagella, not all of them do. Many of them have these pili, and pili are for grabbing onto structures. They kind of help them be sticky and hold on to things. Some pili will allow them to exchange plasmids back and forth, so kind of like sexual reproduction, but not quite, because they're not mixing their genomes together. They just exchange these little pieces, these little rings called plasmids, and oftentimes these carry genes for antibiotic resistance, so very dangerous little things. But we do use them in biotechnology as well, so we can harvest their potential. No nucleus around this DNA, which is circular. It's going to connect end to end, still has the double helix shape if you get down there to the molecular level, but each end is going to be connected. They do have ribosomes that freely float around in their cytoplasm. And since they have no nucleus, as transcription happens, you make mRNA. A ribosome can go ahead and attach to that mRNA and start translation into a protein. So their protein synthesis is a lot quicker. If we look at domain archaea, they also have only one kingdom, the archaeobacteria, and they're also single-celled, but a lot of times we don't see these cells actually leave each other, so they tend to stay together in a little population. Here you can see some archaea that are living in Morning Glory Pool and Yellowstone National Park, and if you see the different colors, these are different species of archaea that live in different temperatures. So the blue can withstand very hot temperatures, the green not as hot, the yellow even less hot, and then the orange and the reds are fairly cool but still pretty warm. So you actually have a living graph of how hot these thermal pools are by looking at the different species of archaea that can live in there. Archaea are also prokaryotic, so they do not have a nucleus. They evolved before the nucleus. Their cell walls do not contain peptidoglycan. Instead, they have a protein sugar complex called glycoproteins in their plasma membranes, and that helps them withstand these hot temperatures better than a cell membrane in a prokaryote or a eukaryote can. So most of the time, they have been associated with extreme environments like these thermal pools, hydrothermal vents like you see here, and sulfur springs, these things. Many scientists don't really use the terms for the kingdoms anymore. It's just bacteria or archaea. Now, I do want to touch on these archaea bacteria that live around these thermal pools. This is one reason scientists think they may have evolved first, because they use chemosynthesis, which would probably have been an easier process for these bacteria to have evolved to have. Now you see here these tube worms that are growing next to these hydrothermal vents. This is the body of the tube worm. This is the mouth. It's not a true mouth, but it's where the fluid will go in. So they take this chemical laden water in through their mouth parts, and it's going to be filtered by these archaea bacteria, and they use chemosynthesis to harvest energy from the sulfur coming out of these hydrothermal vents to create carbohydrates that these tube worms can then process for energy. So a nice mutualistic relationship here between the tube worms, which provide the archaea a home, and the archaea, which provide the tube worms a food source. Domain Eukarya has 4 kingdoms. We have talked about them many times. We have the protists, the plants, the fungi, and the animals. So this is what we call a phylogenetic tree, and it's going to show you how these relationships are assumed to have diverged from your last universal common ancestor, which is here, the first cell. So this first cell going along has a mutation leading to the bacteria group and the archaic group. I don't know exactly which one came first, but the archaeans are the ones that supposedly have led to the eukaryotes. There is a lot of line of evidence for this in biochemical analysis. There's even a particular archaic group that's being looked at as the ancestor, the representative ancestor for eukaryotes. So our archaeobacteria go one way, the eukaryote group with the mutation of having a nucleus is going to go this way. Now, these eukaryotes go along for a while as just one group, and then eventually we split off into different groups. The protists are assumed to have come first and led to and were the ancestors that led to the plants, fungi, and animals, because they have characteristics that fit all of these groups. So we see different variations of the protists that lead us to think that they could have been these first cells that led to these different multicellular organisms. Now, protists and fungi can be either uni or multicellular, but plants and animals are always multicellular. And we're going to look at each one of these kingdoms individually. So you have a nice little chart here to help you with the characteristics of domain eukarya. We do not have the kingdoms of bacteria and archaea on there, but make sure you know those as well. So this is just a general overview and some examples. Now, I'm not going to go into this here, but you can look at this on your own in your slide set, should you need to classify any of these different kingdoms into groups. We'll start with protists. We assume them to be the ancestors of fungi, plants, and animals because they have characteristics of all 3 groups. Protists don't really fit in with any of these 3 groups. They can be autotrophic or heterotrophic, both. Many of them are both. They make their own food and they consume food from other places. Protists are also unicellular and multicellular, so we see examples of both. Here you have a Chlamydomonas. This is a unicellular protist. It's photosynthetic. We see the chloroplast inside, and you have 2 flagella here that help it to swim around. Here you have a multicellular protist called Volvox, and each one of these cells in the Volvox is almost identical to a Chlamydomonas. It's like they're holding together with these 2 flagella. So they have formed a big ball or a group. Inside, you can see a colony of Volvox. This is not a nucleus because this is not just one cell. So this is a new colony of cells that will be put together, and when it's ready, it will burst out of this colony and go live its own life. So it's like this multicellular thing will give birth in a way. So protists They do have characteristics from all groups. They can have a cell wall. They may not have a cell wall. It just depends on what they need to do. Fungi do have cell walls, and their cell walls are made of chitin, which is a very tough sugar that we also see making up insects' exoskeletons. So when you step on a bug and hear it crunch, that's the chitin. So very tough. They do not have vascular tissue, no xylem, no phloem, and they do not have chloroplasts. They don't make their own food. They are not plants. They're very different from plants. Most are multicellular. Some are unicellular. Here you have an example, yeast. Yeast is unicellular, and you can see it splitting off by budding. So many little yeasts can grow off the body of one mature yeast, and then it can break away and go live on its own. But most are multicellular, and we see threads underground that are multicellular fungi. We also see the mushroom, and this is always multicellular. It's used for reproduction. It's the only time you see a mushroom. These little gills here on the bottom are going to hold the spores, which are haploid. Remember that these spores can then go land somewhere and grow into new mushrooms. They can use sexual reproduction, so eggs and sperm come together, temporary zygote that immediately is going to divide, but these are haploid dominant, like moss. They are heterotrophic, so they get their food from other things. They don't make their own. And most of the time, you're going to find them as decomposers, but they can be parasites as well. So decomposers are going to recycle nutrients. Fungi secrete enzymes, special proteins that break down things quickly. And so they secrete these enzymes into their environment to break their food down outside their body, and then they take in the nutrients as they need them. So they're good recyclers. They put materials back into the soil, into the environment. We looked at the cordyceps fungus that affects insects, and that would be an example of a parasite. Ringworm would be another example of a parasitic fungus in humans. And frogs are being affected by the chytrid fungus, which is killing out frog populations like crazy. Now, some fungi do have mutualistic relationships. Orchids absolutely have to have a mutualistic fungi in order to survive and help provide them with their nutrients. We also talked about the mycorrhizae that will connect forests together and deliver nutrients between trees. But fungi live in all environments, not just on the land or in the soil. They can be found in the water as well. Plants are an extremely varied group. They are all multicellular all the time. They are always autotrophic. They make their own food through photosynthesis, and they use chloroplasts in order to do that. Their cell walls are made of cellulose. This is one of the polysaccharides that you need to know. So it's many monosaccharides linked together. Very tough. Used for structure. There are over 400,000 identified species of plants, and new ones are emerging all the time, including everything from mosses to ferns to your angiosperms and gymnosperms as your big trees. This is an example of a rainbow eucalyptus, the only eucalyptus that's found in the northern hemisphere. And as their bark strips away, you see these bright colors. Now, these colors are due to the amount of time that the sunlight has spent hitting this bark. The longer the sun hits it, the more red it will turn, and the new places where the bark has been stripped away are green and blue. So you get the different colors based on exposure to the sun. But they are real and they do look. Animals are always multicellular. They are heterotrophic. They have to eat. They cannot make their own food through photosynthesis, so they do not have chlorophyll or chloroplasts either. They also do not have cell walls. Most of the time, animals need to move around and get their food, so they do not have a cell wall to inhibit their movement and keep them rigid. Now, the first animal cells did not have cell walls, so even your moving animals like your barnacles still do not have cell walls. They secrete a shell, but that's not a cell wall around each individual cell. Most do have systems, interior digestive systems, skeletal systems, cardiovascular systems. They have body symmetry. There are three types of body symmetry. You can have no symmetry, be asymmetrical like a sponge. You could have radial symmetry. No matter where I divide this jellyfish, I'm going to have two equal halves. And then you can have bilateral symmetry, which means you have mirror images. There's only one plane that you can divide a species in half or an organism. So us, for example, we are bilateral. We have a left and a right side. And most animals have bilateral symmetry, which evolved a little bit later. Most species are in the animal group. There's over 1 million different animal species. Some scientists argue up to 10 million animal species, depending on how much of a difference you feel like you need to split animals into different species. Kingdoms are divided into phyla, and phyla require more specific characteristics of the individuals that are placed within them. We're not going to go into phyla in this class. Each kingdom can be divided into multiple phyla, so it would take us forever. But if you do want to get into this, and you want to look at these different classifications, you can look at zoology for animals, plant biology for plants, mycology for fungi, and then microbiology for your protists and bacteria and archaic kingdom, phyla. So phyla, again, have more in common. These organisms are more closely related than just individuals that were within the same kingdom. So the rest of your classification paradigm, remember, dumb King Philip came over for good spaghetti, sushi, whatever. So domain, kingdom, phylum, class, order, family, genus, and species. Getting more and more specific for what our characteristics are as we go down this system. This classification system originated with a man named Carl Linnaeus, who actually contributed a lot to the field of evolution, but this was his major contribution, binomial nomenclature. Binomial means two names. So this is a two-name naming system that he came up with in the 1700s, and we still use this system today. So basically, this is giving every individual species a scientific name, and it has two parts. The first name is always the genus. The second name is always the species. The genus is always capitalized and the species is never capitalized. And these words are always written in italics. So that's how you identify a scientific name. Look for how it is written. A capitalized genus, a lowercase species with italics writing. Most of these are going to be in Latin, which is a dead language, but it's fair to use because no one really speaks it. So this is used to give any scientist anywhere in the world the ability to use the same name in any organism, no matter what they might be working with. So two scientists working across the globe on this particular species of butterfly could call it by its scientific name and know what it is, because this name isn't going to change no matter where you are. The common name, however, will. Now, if we're looking down our classification system, every level is going to have a name. The more we see in common between these species, the more closely related they are. So they're all animals. But if we go down to the phylum level, these two are chordates. They have the spinal cord, and the arthropod over here belongs to a different phylum. It does not have a spinal cord. It belongs to the insect group, and there are a few other members of this phylum as well. So these two, the platypus and this bird, are more closely related than the platypus and this butterfly, or the bird and this butterfly. So that's how you would use one of these charts, and you'll need to use one of those in the future. Notice how these scientific names were written. Again, capital letter, lowercase letter, all in italics, and we see the genus and species written together. We've already touched on this a little bit, but phylogeny is the evolutionary history of a group of organisms, and we arrange this phylogeny on a phylogenetic tree. So this is our assumption of how these evolutionary relationships came about. So here we can see an example of a phylogenetic tree. We start with a common ancestor going along, and you have just one population. Part of that population will develop a mutation and will split off into a different group. The other part of the population may retain those same characteristics and be fairly unchanged for a very long time, or have very few changes. So the split represents a divergence in two populations. And then we see another split here where these two organisms moved apart somehow. There was a mutation that separated these two groups. So these are your common ancestors. The more splits you have to go through, the more distantly related two organisms are. So for example, between species 1 and 2, I have one intersection to connect them together. Between species 1 and 3, I have to pass 2 intersections. So species 1 and 2 are more closely related than species 1 and 3 or species 2 and 3. Because again, from 2, I have 1 two intersections. So that's how a phylogenetic tree works. So branches indicate separations between an ancestral species and a new evolving species. And between those, we have nodes that show there was no change in this ancestor, very little change. The length of a line represents how much time an organism took to evolve or when it went extinct. So for example, this lamprey has a much longer line than this bear. So it had a lot more time in evolutionary history to evolve from this common ancestor than this bear did from its common ancestor here, its most recent ancestor. So sometimes you'll see time put on a phylogenetic tree, but not always. But time is always assumed to be represented. These organisms all have made it to present day. Should you see an organism end early, then you can assume that it went extinct. Now, you can also label these with traits to show what the split may have been. So between the lamprey and the rest of these organisms, we have a common vertebrate ancestor. This lamprey and everything past it all have a backbone. If we continue down this line, we see another split where we have jaws that have evolved. The lamprey doesn't have true jaws. They're a sucker fish. They latch on to fish and they suck the blood out of them. They're vampire fish, basically. But the sunfish, the newt, the lizard, bear, chimp, all of them have jaws. If we keep going, we see a common tetrapod. So all of these organisms have 4 legs. The things that evolved before them did not have 4 legs. Here we have a common amniote ancestor. These are organisms that put a shell or some method of keeping their eggs from dehydrating is used. So the lizard, the bear, the chimp, they use either a leathery shell around their eggs or internal development for their eggs. The newt, sunfish, and lamprey just lay a mushy little egg in the water and it stays hydrated that way. If we keep going, we have a common mammal ancestor. So we see fur, milk production, legs underneath the body, some common characteristics of mammals. None of these organisms have those characteristics. So these are how phylogenetic trees work. There is physical support for these trees, of course, with fossils, and we've looked at the fossil record and how we have these gradual changes that emerge and are retained and build upon. So fossils document how organisms of the past were related to one another, and we can look at physical characteristics with that. Now, sometimes you can even get DNA out of a fossil and compare ancient DNA to modern DNA, which can be fairly useful when you're looking at evolutionary relationships as well. In fact, that's the best line of evidence. Now, the fossil record suggests that all organisms evolved from some ancestral species living about 3.5 billion years ago. And this is where we see our first bacterial mats, very simple organisms. Classification began even before Carl Linnaeus started his 2-name naming system, but taxonomy or these names have changed over time. The earliest classification goes back to ancient Greece with Aristotle, who lived from 384 to 322 BC, so before Christ. And he grouped organisms based on could they move or not, and he put them into groups: plants and animals. Those were the only 2 groups. Now keep in mind, microscope had not been invented, so Aristotle couldn't see these teeny tiny unicellular organisms. He would have had no group for that. We have to wait till the technology catches up before we can figure out more about these evolutionary relationships. So Aristotle's method led to some false relationships. For example, sponges that were pulled out of the sea in fish nets, he would see those as things that didn't move, so they would have been classified as plants. Barnacles, they don't really move so much in terms of getting up and walking around, so they would have been classified as plants. That would have been a misclassification. And again, didn't have the technology for some of our other kingdoms like protists, bacteria, and archaea. Now, Linnaeus did develop his hierarchy system based on body shapes. Again, he could not analyze DNA. He did not look at embryos. So bats could fly, so they were related to birds. False classification, because we did not have these other pieces of evidence that were looked at, like their bone structure and whether or not they had fur versus feathers. So kingdoms were his largest level of classification. He did not look at domains. Again, he would not have even seen domains, bacteria and archaea. So he would have only had one. Now, we still use classification, plants and animals, but it has changed since Aristotle's time. So the system has changed due to advanced technology. Microorganisms were discovered after the invention of the microscope. Prokaryotes weren't able to be divided into kingdoms at all until we could chemically analyze the composition of their cell walls. Bacteria made a peptidoglycan, and archaea has glycoproteins. But with just a microscope, we see those as being the same kind of organism. They have a lot in common. They're both unicellular and live pretty much anywhere, though archaea are more associated with extreme environments. Fungi used to be classified as plants because they didn't move and they kind of look, kind of like a plant. But we know now that they're heterotrophic and have characteristics more similar to animals in their chemistry. Now we can even use DNA strands to look at for classification using gel electrophoresis to separate these fragments and make these DNA fingerprints. So remember that our DNA is loaded into this gel, it's exposed to electricity, and that DNA runs down the gel. Big pieces stay behind and small pieces travel further. So the more similarity we see in a fingerprint, then the more closely related two organisms are. We have tools that we can use to identify organisms called dichotomous keys. And dichotomous keys use these characteristics to separate organisms into groups until you get down to your species level. So di means two, and these are pairs of questions, either basically yes or no. Do you have this characteristic or do you have this characteristic? So two questions and each question will lead you to either a new question or the answer to what your species is. No matter what species you look at, you always start with question one. So we'll look at an example here with very simple dichotomous key. The first question, do you have 5 petals or do you have 7 petals? Now we are counting the petals. So 1, 2, 3, 4, 5, 6, 7. And since it has 7, it led us straight to the species, which is the starflower. You see its genus and species here, capital letter, lowercase letter. Okay? Very simple one. Let's go to one that takes a little more effort. So we'll analyze bird X here in this dichotomous key. We always start with question one, even though we're not starting with the first organism on the list. So bird X, either the beak is relatively long and slender or stout and heavy. So this isn't a very long beak. We can say that it's stout and heavy. So that tells us we need to go to question two. The bottom surface of the lower beak is flat and straight, or the bottom surface of the lower beak is curved. So this is the bottom surface of the lower beak. We can see that it is curved. We follow that and go to question three. The lower edge of the upper beak has a distinct bend, or the lower edge of the upper beak is mostly flat. Well, if we look at the lower edge of the upper beak, it is flat, and that leads us to the Platyspitza, which is the genus of this bird. And we know it's the genus because it's capitalized. That is all for classification, and that's all for your EOC for the time being. So if you have any questions at all, then please contact me. Otherwise, go ahead and complete your quizzes on this subject and get ready for that EOC, which is coming, and look for those review activities. All right, have a great day, guys, and bye-bye.